Get started, and I'd like to begin by welcoming the Danes. It's been a long time. We've been working with you and your family since I think 2002, and so I think fast forwarding 13 years, and everybody can hear me okay? It's 2015, and that illustrates at least one thing about scientists and the scientists in my lab. If nothing, we are persistent. And by being persistent, we've been able to solve or identify the genetic cause of hearing loss that's running in your family, and the scientists responsible for this kind of enlightenment are Dr. Ella Aziz, who will be presenting, and Kevin Booth, who was an undergraduate here and has worked in the lab all during his undergraduate years, and we've been able to attract him as a grad student, and so he'll continue in the lab. And their perseverance and their enthusiasm and passion for science is an illustration of the quality of people that I'm fortunate enough to have in my lab, and also the fact that there will be a legacy after I retire in 15 or 20 years of persons who work really hard and are incredibly brilliant. So you're probably wondering what this picture is and why I am showing this picture, and it's actually an illustration from The Mysterious Island. And The Mysterious Island is a book that was written by Jules Verne, and it was a sequel to 20,000 Leagues Under the Sea. And probably all of you read or saw the movie 20,000 Leagues Under the Sea. I saw the movie, and I remember I was a little boy, and I was scared when the giant squid came up and got the boat on the surface. And I probably left the theater at that time. But anyway, the Mysterious Island, the secret, is revealed when it's discovered to be Captain Nemo's hideout. So he sails in the Nautilus all around the world until all of his crew members die, and he's the only living crew member. And then he goes to the Mysterious Island, and that's where he keeps the Nautilus. But what's interesting about this picture is that the picture contains a lot more than what you initially see. And I show it as a reflection of the science that we do and the fact that you need persistence, but you also need to look at things in unique ways and different ways, because by so doing, then sometimes you see the unexpected. And so this is an example of what's called anamorphic art, where if we put a reflecting mirror over where that sun was, we can see the picture, the portrait of Jules Verne. And so with that, I'm going to turn the podium over to first Hala and then to Kevin.
Thanks. First of all, I would like to welcome all of you, and we are happy to have you here. It has been a journey working with you, with your family, to identify the causative gene in your family, and I would like to walk you through this journey with us. So, I will just give a brief review about the hearing process, the history of deafness gene discoveries, that's very important. Then we will start the real stuff, what we have done with your family, with your sample DNAs, the methodology we use, and how our methodology, our way of analyzing the data, our way of interpreting the data has evolved in parallel with advances in technology and our knowledge of the hearing mechanism. And then the most important part, we will get to the findings. What did we find? And how this will build and to develop more therapies, and what we are going to do with that, with the knowledge that we gained today. So the ear is composed of the outer ear, the middle, and the inner ear. The way we hear is, if we have a cow singing, for example, this vibration or the sound will get till the middle ear, where it is going to provoke the vibration of these small ossicles. This vibration will be then will travel then to the cochlear. That's the cochlear, where it is going to be translated into electrical signals that will travel through the nerve to the brain. And that's when we understand what is happening. So from there, anything that you can clearly see that anything that goes wrong throughout this whole process here will result in some kind of hearing loss. So hearing loss is the most frequent sensory disorder in the world. Currently, there are 360 million people affected with hearing loss. And we predict that by the year 2020, there will be over 1 billion people affected with this. Hearing loss could be due to environmental causes, like viruses or some antibiotics. But in the most part, it is genetic. So the cause is in our genes. And for genetic causes, we classify them into syndromic and non-syndromic. So non-syndromic means there is only hearing loss. This is the only phenotype that people present. Syndromic is they have hearing loss in an association with another disease. Like for Waterberg syndrome, there is a pigmentation. There is a pigmentation, another pigmentation phenotype. For Usher syndrome, they have retinitis pigmentosa, which is blindness. For Bender syndrome, in association with hearing loss, they would have water and some problem with thyroid. Amongst the non-syndromic, they can be classified into recessive and dominant. Recessive is when both parents are normal hearing, but the kids have some kind of hearing loss. And in this case, we expect each kid to have received one muted copy from each parent. And that's why it's only when they have 2 copies from each parent that they develop hearing loss. For dominant, it's different. For dominant, one of the parents has to have hearing loss, and it will pass it over to his kids. So till now, there are over 160 genes that have 106 genes, that have been identified as causing for hearing loss. That is extreme. That's a lot of genes. Here is a history, a little bit of a work through a history of gene discovery for hearing loss. It actually really took off in 1997 with the identification of the first gene, GJB2. Since then, as you see here, there are a lot of genes each year that have been identified. These represent 106 genes. The ones in red are the ones that our lab has identified. So we contributed to the identification of 20% of all known deafness genes.
Wow.
Thank you. So here I also added a few milestones. Your family actually visited the lab first time in 2002. And in 2003, this was the first time that the Human Genome Project was achieved. What that means is this was the first time that a whole genome of one person was fully sequenced. And at that time, it cost $3 billion. Things evolved since. In 2008, this was the first time that whole exome sequencing was performed. What whole exome sequencing means is it is the sequencing of every single gene in the genome, and there are 20,000 of them. And this was possible by the advancement of technology, what we call next generation sequencing or high throughput sequencing, which means we can sequence all of these 20,000 genes at once, at the same time. The cost of whole exome sequencing in 2009 was around $9,000 per person.
Wow.
The year after that, our lab developed the first comprehensive genetic testing for deafness in the world. And in 2012, that's when it was streamlined as a diagnostic test. And it cost $1,500. Oh, sorry, yeah, it's $1,500. The whole exome sequencing price in 2014, again, this is per person, cost $4,000. And in 2015, that's when we solved the Dane family genetic code, and that's priceless. So let's talk about a little bit about your family. So we had DNA from Mr. and Mrs. Dane. We have DNA from Jim, Ruth, Margaret, Bill, Donna, Harold, Helen, and some of the grandkids. This is the audiograms we have. And here, if we look at the audiograms, we can see that they have different severity and they have different shapes. We can see that James, for example, has hearing loss only in the high frequencies, the same as Helen. But Margaret and Donna have a more severe to profound hearing loss. And we will go back to that. And that actually adds another level of complexity to the hearing loss in your family. So when you came here in 2002, at the time, as I showed you earlier, the technology wasn't that great. That's what we used to use, and that is what's time consuming. At the time, the only available test was for GJB2. This was the first gene identified for hearing loss. And we screened that, and it was negative. But science is a lot like farming. How is that? So it's all about the tools you use. In 2002, we were using this tool. It is comparable to these kind of tools. So these tools, they are, to get anything, it's laborious. It takes a long time and a lot of effort. And the yield is not always great. That is the same with this novel technology now that has been developed. This is the modern novel technology, which is high throughput sequencing. And you can compare that to these huge machines. So they make things fast, it is high throughput, and the yield of product is huge. It's great. Let's now switch a little bit to the genes and what have we used this technology for. So if you think about the genes, each state in the United States has one gene that is responsible for hearing loss. So Iowa would be GPR98, that's one gene. And we do not really screen all the gene because there is only certain pieces that are important in this gene and that are worth screening with our current knowledge of science. So Iowa has 99 counties, and that kind of represents, and GPR98 has 90 exons. These exons are the little pieces, so it is comparable. So the way we used to do it before is we are looking for something abnormal in all these counties. So we have to start by 1, it's not here. We have to screen all of Johnson County, it's not here. Then we have to go to the next county and see, it's not there either. We have to do it one county at a time. And we were not able to go to the next county till we make sure this one is not the real one. Now, if we compare this to the novel new technology, that is kind of taking a snapshot of the whole solar system at once. So we are going to look at all of this, all the planets, all the stars, everything that is in there at once. And then we can, from that snapshot, we can look at a specific place to start where to look. We can look at the Earth, and the Earth would represent whole exome sequencing. So it's only what I saw here, it's the whole genome. It's coding regions, important regions, less important regions, we don't know what they do, what they are doing, or what's their importance, but we screen them anyway, we sequence them anyway. The thing is, we cannot analyze that much data, it's too much. So we can focus in just one thing, planet Earth. Planet Earth in this way would represent exomes, to represent the 20,000 exomes I was talking about earlier. OtoSCOPE would represent only the United States. That is 50 times. So the United States size is 50 times less than the Earth. And that would represent, instead of looking at the whole Earth, all the continents, the subcontinents, all the oceans, we are only going to focus on the United States. And that's what we are doing with OtoSCOPE. OtoSCOPE, it is the test that we developed. This is the high throughput comprehensive genetic testing for deafness. And we are the first one, the first lab in the world actually that offered this. That developed a test that using high throughput sequencing. Yes.
Now, when we were here last time, you had developed the OtoSCOPE.
Yes.
And you were using it. Okay.
Yes. So this is a great question. Why? Because there are different versions of OtoSCOPE. As I showed you in that timeline for novel gene identification, you see that each year there are novel genes that have been discovered.
Right.
So we developed version, let's say now we are version 6 of OtoSCOPE. We have the version, we are working on it. But meanwhile, other people or us, we also identify novel genes. They are not in V6 yet, but they will be in V7.
Okay.
So that's how for example, we run some samples on version 5 or let's say version 5. We didn't find any mutation in that family. We ran them in V6, we found it. That's because the gene was not in V5, it was in V6.
Okay.
So with the current version we have of OtoSCOPE, it's screening all of the genes. It's screening 106 at the same time. And I want to emphasize that your family was actually one of the first few families that we ran on OtoSCOPE V1. So, what is OtoSCOPE? This is just an overview. So we got blood samples from you, and we extracted DNA. That takes around 3 days to get the DNA. DNA, we did some bench experiments to make the libraries. That would usually take 7 days. After these 7 days, we ran into, I showed you this one earlier, this is the high throughput, really new cool tool. And that takes around 2 weeks to run. It only becomes actually very exciting when we get the data back. As I said earlier, since we screen so much and so many genes at the same time, the amount of data we get is enormous, it's huge. And then manually we cannot analyze all this data, it is impossible. So we rely a lot on informatics and bioinformatics. And we have been working for the last 5 years. We have been working for the last 5 years on developing these new softwares, new pipelines that makes the analysis faster, it makes it more accurate, and it makes the results easy to analyze and to understand, actually, because that is a big part of the whole process. After we're getting the analysis done bioinformatically, that's when we meet. We have a weekly meeting, we call it hearing meeting, and we meet in this meeting, we have clinicians, we have scientists, we have geneticists, we have bioinformaticians, we have technicians, we have genetic counselors too. What we do during this is we are going through the data that came out of the bioinformatic analysis and trying to make sense of all these variants. These variants are all the differences that we found from the sample genome when we compared it to a reference, to a normal genome. So, but here we obtain a huge amount of data. So we need to do some kind of filtering to finally narrow it down to only the most important variants. And that process is a lot like running for loans. So it is a laborious process. It is very tricky, but the results are golden. So what do we do, or how do we do to filter, to get through all these variants, make sense of them? What we do is actually we need to check if this variant, this difference, this mutation, or is real. Because sometimes with this technology, as we said, we sometimes get false positive, which means it is not there, it's just because there is a glitch, there is a bug somewhere in the machine, and that's why we get these errors. We need to make sure that this variant we found is of good quality, because if it is bad quality, we cannot trust it. We need to have stuff that we can trust, because everything downstream is based on that. Then, that is the filtering. So then what happens is, has this variant been found in other normal hearing people? Because if we found this variant in normal hearing people, it doesn't make sense. That is the 41. And then is this variant is actually in a gene, in these pieces I told you about that are really important, the exons, because that would make more sense that they are causative rather than are somewhere that we don't need or the genome is not using. And then is this variant consistent with the family history? As I showed you earlier, if it is a recessive family, we are expecting each affected individual to have 240 or 2 mutations. If there is only 1 mutation, that's not consistent with recessive. If it is dominant, we are dominant family, we are only looking for 1 mutation that is transmitted from one of the parents. Finally, we got to this variant list. These are very good candidates. We always end up with more than 1 variant. And we have to go through all of them kind of to assess how strong of a candidate they are. So to do that, and that would help us to come up with a diagnosis. But it is kind of a piece of the puzzle where each piece has to fit to make a diagnosis. So the variant frequency, as we talked about, how frequent is it? Because sometimes even if it is seen in the general population, it's okay. If it is recessive, it means if this person has only 1 copy, 1 mutant copy is still okay. The disease only appears when there are 2 mutant copies. The type of variant, is it a stop mutation? Is it like how severe or how pathogenic this variant is? Pathogenicity prediction, that is, we use a lot of software, and our lab has worked over the years on developing new software to see, we give them this variant and let the tool or the software work, and it will predict if this variant is very important, and if it is, when we change it to something else, it will actually be damaging to the protein and to the gene. The functional data, has anyone before worked on this specific variant to see it really affect? Like they studied in the cell, they saw what it did in the cell. The phenotype correlation, sometimes we see mutation in a specific gene that is supposed to be causing severe to profound hearing loss. But what we see is progressive, starting in the late 30s, and it is downsloping, like it's only moderate hearing loss. So that does not fit the phenotype. And the most important, also, one of the most important pieces of this puzzle is the segregation, which means if we have a family, every single affected person in this family has to have this mutation. If he doesn't or she doesn't, it means it doesn't segregate, it doesn't make sense. And only then, when all the pieces fit, that we can have a diagnosis and on the genetic dose. So as I said, each step, what I showed you earlier, we have been really working hard the last several years to develop these new tools. One of them is the deafness variation database, and this is a tool where we put our lab in collaboration with the engineering department. And this tool is really important not just for scientists or researchers, it is also important for clinicians, because when they get results from their patient, they can go there and check if these variants, if these genes have been involved in deafness before. We have also developed, and it is, as I said, we have been working really hard on this kind of underlying mechanism and process. We have also developed AudioGene, and this is also trying to correlate the phenotype to the genotype. So any one of you now can enter and put their audiograms. It's really straightforward, and it will predict what kind of gene is predicted to be responsible for your phenotype. So it is prediction, but we have based it on a lot of study that we have been doing for over 5 years now. So using OtoSCOPE, this is the solve rate for OtoSCOPE, which means how many people, what's the percentage of people we were able to diagnose over the years. And you can see here that the solve rate depends on several parameters and factors. One of them, if it is dominant or recessive, we have more success with dominant than recessive. But the overall solve rate of OtoSCOPE is a little over 40%. why is it only 40%? Why not only, why 100%? Why aren't we able to solve the other 60% of these families? The reason is there is still so many other genes that we did not discover. There is a lot of them, and these families or these patients might have these mutations in these unknown genes. So if we go back to your family again, so that's what we have. We have run your family in 1, 2, 3, 4 different versions of OtoSCOPE. We started in 2010 and in 2013. And the colors represent, we ran some of them several times, and some of them just trying to figure out what do they have in common, what is the genetic code. We ran the first analysis, we only ran one sample, and we didn't find anything that made sense. We then ran V2, version 2 of OtoSCOPE, 2 more samples, it still didn't make any sense. We didn't find anything that was significant. We did it on 3 samples with V3, and we still, the code was still elusive. And then we did also 2 more, and then it did not make sense at all. So from that, what we say is, we were looking maybe at the smaller set of genes. As I said, OtoSCOPE, it's kind of only known hearing loss genes. So why don't we go and look at the whole exome? And for that, we ran six of them, both Mr. and Mrs. Dane, and all the kids on exome sequencing. And because we suspected that the genetic cause is going to be a little more complex, we based that on, we had 4 different kind of hearing loss that are different, and that might be the reason why we suspect there might be more than 1 gene. So maybe these 2 have inherited 1 gene, 1 mutation from the father, where these 2 inherited something else. We tried, maybe actually they inherited, it's a dominant, we are still assuming it's dominant, because here we thought this one is genetic codes. So maybe actually what they received is from the mom, not from the father. It still didn't make sense. Maybe they received all the analysis. We did a lot of analysis, all the possible combination that we could think of. Maybe actually they received 2 different things from both parents, and that's what made them 2 have more severe phenotype than the other ones. Still nothing. So we took a pause just to think, reassess everything, and as Richard showed earlier, we had to think about it in a different way. We had to change perspective, kind of sit back and look at it again from a different angle and really think about it. So what we did this time, we just assumed that both parents actually do not have a genetic cause of hearing loss. So their hearing loss might be due to its age-related hearing loss, might be due to noise-induced hearing loss, the environment, as we saw earlier, 20% of all hearing loss is due to environmental causes. And then when we did that, actually what we did, this is to show you the whole exome thing. For each, when we did whole exome sequencing in both Mr. and Mrs. Dane, we found over 200,000 variants. This is 200,000 variants that are different from the human genome. The reference in human genome. That's a huge data.
We knew they were weird.
That's a lot of data and a lot of variants to go through. So as we saw earlier, we had to do some filtering to kind of come down a little bit with these numbers. So after our filtering, we got around 3,000. That's a little manageable. What happened then is the kids have to have a specific combination, because what we are expecting here is each one of these have received a specific variant from the father and specific variant from the mom. So here we have different combination. I'm only representing 6. So each ball here represents one variant. So can any one of you tell me if these kids have the same pattern or the same combination? Which balls do they have the same combination for?
Like the fourth one down.
Okay, that's one. Yeah. So which is the third and fourth? So this is only 6 variants. Imagine how we are what we deal with when we have 3,000 of them. We have to go through every one of them. Don't get me wrong, it is fun, but it just takes a long time. And that's how we found it. Following our this strategy, we actually found 2 mutations in TMCI gene. So how we did it? We did Sanger sequencing, and as you can see here, there is mutation 1 that comes from the mom, and the mutation 2 comes from the dad. And if we look at all here, they all have the mom mutation, the dad mutation, all of them. Harold doesn't have any of them.
Wow.
And if you look at the kids,
It's not.
if you could have, that is lucky, yes.
Yeah.
If you look at the kids, these two have inherited the variant from their grandfather, and these two inherited the variant that came from their grandmother. So that was really, really exciting for us. We were actually running in the Kevin and I, it was late at night, we were working on it, we got really excited that we couldn't get home because we wanted to really figure it out. And we were kind of just yelling and screaming and running down the hall in the lab. Yes, we have it.
We met.
Yes, exactly. It was a fantastic time. It is one of those moments that really keeps us going. It's just to get one of those moments again and again.
Okay, I'm confused, though. Can we stop?
Yes.
Just take a little break. Okay, so the red is a variant from Allie, from the mother. And the blue is a variant from John.
Yes.
Okay, and so then that went down, let's say Peg is the second one, the Margaret.
Yes.
Okay, all right, so.
So Margaret here received this variant from her mom
Mom and her dad.
and the other variant from her dad.
Yeah,
Yeah.
and then.
It went.
And then she married someone else, and then her kids, they have to inherit either this one or this one.
Oh.
Because the 2 copies that Margaret has are both mutant.
Oh.
So
I
no
see.
matter what, her kids are going to have one of those, either that or the other one.
Oh, okay,
And so that worked out. The pattern is,
Yes,
that's what confirmed.
yes.
I see,
So
okay.
I'm the third one who didn't get tested.
Okay.
What's my odds of one or the?
Well, you're gonna have to have,
You're Am I probably earlier? Just
So, where are you in here?
nowhere. I'm
Right.
Here?
no,
I'm with the 2 ones.
Okay,
Yeah.
so you will have either the blue or the red, and heterozygous, which means the other allele would be normal.
Okay,
Yes. So you will be kind of either one of these or one of these.
okay.
And we can test you if you would like to. Yes? Any other questions?
well, we have a daughter, maybe we should have her test.
Yeah, you are welcome, anyone. Yes?
Another question.
Yes.
So I have 2 daughters. What's the possibility of them carrying that down?
So you have 2 daughters, which means they have 50% chance
Okay.
of receiving the mutant allele.
Okay.
One of them does. Yeah. Yep. Any? Yes. That's a great question. I will get that. I will get to that in a couple of slides. So when we found these 2 variants, we just needed to check a couple of few more things. Just it's kind of validating, making sure everything fits. So when we looked at, remember when I said it shouldn't, the mutation should not be present in a huge a fraction of normal hearing people. So here, for mutation 1 from the mom, this mutation was found only 9 times in a million
Wow.
alleles. The other one coming from the dad is found in 1 in 1200, which is for recessive is still good. Now, if it was dominant, that's another story. Because when we say dominant, even if you have 1 mutant copy, you will have the disease. For recessive, since you have to have 2, so the chance or the probability is going to be lower to have them both at the same time. One of the things to check if this variant is important is if it was that important for the function of the protein, it should be conserved across evolution. Because all these animals have the same gene. Every one of these have TMC1 because it's involved in, I'll talk about it later. But we checked, and yes, even the fruit fly has the same exact amino acid at that position. So to come back, what is TMC1? TMC1 is for transmembrane channel-like 1. Its function till now is controversial, and it's not really completely understood. Some people, what we know is it is involved in mechanotransduction. What is mechanotransduction? If we go back, you remember when I showed you this earlier, this is the ear. This is the inner ear, or the cochlear, we call it. If we take a section and zoom in into this part, we will find this little thing here. This is the organ of Corti. We zoom in into this, that's what we have. And what we have here is 3 rows of outer hair cells and 1 row of inner hair cells. These cells are very important. Why? Because they are the tool for mechanotransduction. We call them actually hair cells because if you see here, they have little hairs on top of their head. So if we zoom in here, that's what we see. This is the hair. And then here is a zoom-in image. If we zoom into this little thing between 2 hair, we call them stereocilia or projections. If we zoom in there, that's what we are going to see. This is 1 stereocilia, this is this one, and then the second next to it. What TMC1 does, that's where it is localized. What people think it does, we know for sure it is involved in mechanotransduction, which means it is involved in translating the mechanic action from the sound vibration. It is, they are translating it into electrical signals that go to the brain. And what it does here, some people say this channel here is the one responsible for the mechanotransduction. Some other people think it is just associated to the channel that is involved in mechanotransduction. It doesn't matter. What we know is it is important, it is very important to this process. And without it, there is imbalance in calcium in the hair cells, and which at the end will make these cells die and degenerate. And that's how we have hearing loss, because these cells cannot survive in this environment. So, does anyone have a question? Because otherwise I will let Kevin talk to you about what is to come. Good. I'm glad everyone understood that.
Hello, can everyone hear me? Okay, perfect. So, as Hala just explained, that everyone has the same cause of their hearing loss, but yet we can clearly see that there are different results. And we're very interested in to know the why. And it kind of falls into this idea of expectation versus reality. We expect everyone to have the same type of hearing loss, but in reality they don't. And so we had to ask ourselves a very important question. What is the normal hearing loss? And what I mean by that is number 5 and number 4, is that the actual hearing loss that should be caused by these 2 mutations, Or is number 8 and number 3 the actual hearing loss that should be caused by these mutations, and there's something else going on that's protecting number 4 and number 5? And so what we consider this is when we have 2 different phenotypes or 2 different types of hearing loss that happen to be in the same family, we refer to that as an outlier phenotype. And now there are 3 hypotheses we have for why this is occurring. The first one has to do with the amount of TMC1 that is inside your bodies and inside your hair cells. The second one has to do with the TMC1 interactions and what is in the neighborhood of TMC1 that also could be affected. And then finally, we have to look at the actual mechanism where TMC1 is to see if there's anything else in that area that is affected. So first we're going to start with the amount of TMC1. One of the mutations you have is what we refer to as a splicing mutation. And simply what this means is, like Hela said, in our genes, we have sections that are just not important, and we can just get rid of to create a proper protein. And so I like to think of this process as, does everyone remember when you were kids in school and you had to cut on the dotted line? And if you didn't cut on the dotted line, then your picture was either too small or too big. This is the same idea, is your body cuts out these sections and creates the appropriate picture of what the gene should be. Sometimes, though, like in your situation, you have a mutation in that section, what causes the wrong cutting to happen, and therefore the wrong picture should be created of what your gene should look like. There's also, though, a little bit of variability in this process, meaning it's not absolute. You don't have 100% of this or 100% of this. There's a wide range that can happen. And so, And so what happens is we can have the mutation, and we can still get normal protein and in the normal gene picture, but we can also get the abnormal picture as well. And so with this, what we think might be happening is that each one of the children have a different amount of the normal gene. So for example, we would have number 5 and number 4 would have 25% of the normal gene and 75% of the mutant gene, whereas number 3 and number 8 actually has much less normal gene, which inhibits the function of the protein. The second thing I want to talk about is TMC1 interactions. And we can do this now, and this is a very new technology that we have by looking at pathways within the genome. And so basically what we can do is we can ask ourselves, what does TMC1 interact with? What other things affect TMC1? And we can see here that there's only a few, but this isn't a very accurate picture of what's going on. And so I want to use this analogy. Let's say you want to sell your house, and you want to figure out what is the best value to sell your house for. You can either look on the street, the same street you live on, to appraise your house that way, and that would be looking like this, is looking just on the street to figure out what the value of your house is. But realistically, you should actually look in your neighborhood to look at to figure out the value of your house. And so that's what we have to do, is we can't just look at the 5 or 6 genes that are interacting directly with TMC1, but rather we have to take a more global view, and we have to look at the whole entire neighborhood. And then finally, I'm going to go back to the image that Hela just showed you of the tip link. And we're going to look at the different protein interactions there. So this is the picture, and again, here's TMC1. And there are a dozen proteins here in this picture alone, which all are essential and are needed to work. So if any of these proteins are are mutated, it can be either positive or negative, and it can cause further problems with TMC1. And so it can either help with the hearing or it can be detrimental to the hearing process. So those are the three very overview reasons why there's a difference in the phenotype. However, this issue gets compounded when you start asking that question again of what is the normal hearing loss? Because you have to ask yourselves, is what's going on is due to protection versus something that's being detrimental? You also have to ask about the inheritance. Hala showed you many slides in which you considered all the different inheritance patterns for the hearing loss. And finally, we have to ask ourselves, do we have the right filtering process? As Hala mentioned, we spent the better part of half a decade trying to fine-tune this process. Right now, what we're doing is completely novel. And we don't have a blueprint in front of us. So every time we fine-tune anything or we alter something, we have to start all the way back again at this point. And we have to start and we have to look through every single thing again. So probably right now, it seems as though we're in the dark. And it's kind of, we don't really know where we're going. But there is light at the end of the tunnel. And in our lab, we are very excited about seeing this light for two big reasons. First, there's actually proof that there is a genetic modifier for the gene that is causing deafness in your family. Recently, a lab just published their results and which show they have a mouse with a mutation in TMC1 with hearing loss. When they breed that mouse with a normal hearing mouse that has a mutation in an unknown gene, the results of that is a mouse with less severe hearing loss. That is very, very similar to what is happening in your family, where you have two individuals with much more severe hearing loss, and they possibly could have inherited a gene that is protecting them and causing their hearing loss to be less severe. So this is very exciting work, and as this proceeds, our analysis and understanding of your family in particular also will advance. Next, I'm going to just briefly touch on gene therapy. For over a decade, our lab has been pioneers in trying to solve the problem of hearing loss, not by implantations and not by hearing aids, but rather on a molecular level. And so what we're doing is we're taking a mouse, which actually is the mouse that has the gene mutated, which is the same in your family. It's a different mutation, so the results I'm about to show you are not directly translatable, but it's the idea that it's a proof of principle. And once we get this worked and once we iron this out, then we can open the door and we can look at specific mutations, such as the ones that are in your family. And we have several different mixtures and concoctions going on that we've tried throughout the years. And very recently, we discovered that when we inject the right one into the mouse's ear, and then we test the mouse's hearing, very similar to the way we test humans' hearing, what we find is we find that we can improve their hearing. So this is a very busy and colorful slide, so I'm going to walk through it with you. On top, we have the mice that have no mutations whatsoever, and they have what we consider normal hearing. In the blue and green, we have the mice which have the mutation that did not receive any type of treatment. So you can see between the non-mutated mouse and the mutated mouse, there's quite a bit of difference. And then what we have in red here, which we're very excited about, is the treated mice. The mice that have gotten this treatment, and we've been able to preserve their hearing loss. We've been able to preserve their hearing. Now, these results are only for about 8 months out because they're very new, and this is ongoing. And so, but we're very excited about these results, and we're going to continue to gather data further out to make sure that this is viable, and we're going to continue our studies on this. But we're very excited.
So when you treat them, are they alive and then you inject something? So
Yep.
then and their hearing improves.
Yes, so it's kind of back to the to what Halo was explaining, was that the cells inside your ear, they die off,
Okay.
and so therefore you have the hearing loss. And what we're doing is we're actually correcting the mutation that's found in the mouse, and their cells inside their ears are not dying off based on our data.
So is this more like protection and mode then, instead of?
Yes, so this would
You're
this,
not
this would-
generating new.
No. So that's another area which is very hot in this field right now, is if we can regenerate, then that really might be the way to go for people who all their cells have died.
But this right now is protecting it, like you
Yes.
do this like in an infant. that way
Yes,
you-
exactly.
Okay.
Yep. This would, yes.
Anecdotally, it seemed that Donna always had very severe hearing loss from a very young age. Peg did not have as severe a loss, I don't think, when she was born, because she can carry a tune, she can sing, she was she went out for chorus, but I'm gonna stop there. But Donna doesn't have a sense of tone that Peg has. And even Jim's hearing, I think, is much more severe.
It's getting severe, right? Progressive, it seems.
And not just age-related, as we talked about the last time we were here, where you can project it out. It just seems like there's just that much more substantial fall off, and Helen even more
Especially
so,
Helen.
because Helen is really, she never had what anybody considered a hearing loss through most of her high school years. And now it's pretty severe. So anyway, but what you're saying is you inject them before the cells die off.
Yeah, so we are injecting the mice at a very, very young age. And so it's right about the time the hearing process starts within the mouse. So, yeah. Any other questions? Okay, and so with that, I just want to conclude.
I had a question from your prior presentation too, as to when you said we've identified these genes here, is the one that you're talking about here a new identifier for deafness, or one that was previously identified?
This gene has been causally implicated maybe for 5, 6, 7 years. The issue has been that we have never looked at your family the right way. And so the evolution in time has been in part in our thought process, allowing us to look at the data a different way. That was one thing. The second thing that we did is we then looked at all of the data blinded, so irrespective of the degree of hearing loss in your family. And it was only by doing that that we could arrive at this conclusion. This is a real solid conclusion because those two variants are extremely powerful variants as causes of hearing loss. And so the golden nugget in all of this for us right now is the lab is currently spending a lot of effort on modifier genes. So if we could understand why 2 of the Danes can hear better than the other 2 Danes when all 4 carry the same genetic mutations, that opens up a possibility of a different type of therapy, because if we can understand that mechanism, we should be able to apply it to others with this kind of hearing loss. And yours is actually the only family that we have, so yours is a recessive family. Yours is the only family that we have, and we have thousands of families from across the world in which we have this particular paradigm. We've got a couple of dominant families now, and so we're actually trying to collect these families because, as you can see from that mouse study, there are genetic modifiers that can affect the degree of hearing loss that a person has. And so this potentially can give us insight as we move forward to develop different types of rehabilitation options for hearing loss.
When you say that you just determined that needed to look at the data in different ways, is that analysis transferable for other studies?
Yes,
It's not just applicable to this family.
no, it's transferable to other studies too.
So how soon can we start treating our kids and our grandkids?
You don't need.
And make sure that they don't have, do that protective thing?
You won't need to treat your kids and your grandkids, but one of the questions your kids and your grandkids might ask is, what's the probability that in marrying somebody, I will have children with hearing loss? And we can answer that question for them based on the frequency of the variants in these genes that cause hearing loss. So we can provide meaningful genetic counseling if they want to do that. We can do the genetic testing if they want. That's real easy. We can do it on a saliva sample.
Is that something to come in and do, or? done
We can
from
even
home?
mail it to you.
It's,
You could treat mice at a young age, and that would improve their hearing loss. What about treating mice, older mice? And my question is, what can you do for these 4 kids now to help their hearing?
Yeah, John, I don't think we can do anything right now. So and that's unfortunate, but we're still trying. So what we have been really focusing on is what we think might be more feasible. And we think it's more feasible to prevent something from breaking than to fix it after it's broken, kind of thing. And so we've spent a lot of energy, as you mentioned, preventing the hair cells from deteriorating. So maintaining hearing when, if it wasn't for our intervention, you would have hearing loss. And we're getting really good at doing that now. As far as restoring hearing after it's gone, there's some work that some labs are doing. I think that's a little bit further down the road.
so, in other words, 33 years ago when I was pregnant, and I called up, and I did call the university, and I said, do you have any predictive tests for hearing loss? And they said, no, not at this time. Now, my grandchildren can do that.
Again, we've actually
Wonderful,
just,
yeah.
we've been compiling the data, and it turns out that after an audiogram, the single best test that you can get to predict, for example, hearing loss, the rate of progression of hearing loss, anything else associated with hearing loss is genetic testing.
Wow, that's great.
And that's.
And then there might be, if there is this variant, that they could have, maybe in the future, some.
We like to think that
Preventive.
this is laying the foundation for other therapies, so that maybe we can put Bruce Gantz out of business.
That's wonderful.
So are you all authoring an article that's going to be published?
Well, we haven't quite got that far. This is about as far as we've gotten, just sharing it with you.
Wow.
So, I feel like we've kind of danced around this, but just ask it directly. So for, I guess my sister with her 2 daughters and JJ and her daughters, is it in their best interest to have them tested so that therapy can be done at a young age to prevent possible hearing loss that would be attributed to this genealogy?
So they will not get hearing loss attributed to these genetic causes because you have to have 2 variants.
They're okay.
But we can test them if they would like to know if they're carriers, just like their grandparents.
But we don't know that, do we? If her husband is a carrier, then they could have.
It's possible.
Right. You're right. So, for example, her husband has a 1 in 1,000 chance of carrying that splice site variant just at random. And so then, if you were a carrier, you have a 25% chance of having affected children. So there's a 1 in 4,000 chance that you could have a child with hearing impairment due to TMC1, just based on probability. And we can actually refine those numbers if you want by just doing the genetic testing.
So when you said that you are getting good at helping keep people from losing hearing, not necessarily with the restoration of lost hearing, what are the therapies that can be offered to keep, the hearing loss from being more profound if it's already started?
Yeah. So we're spending a lot of effort on dominant hearing loss, and we're spending a lot of effort on, so with dominant hearing loss, you have a variation in one copy of the gene that is causing the hearing loss. We're spending a lot of effort on therapies to prevent that copy from being expressed, and if we can prevent it from being expressed, then you have normal hearing. And that's what we're trying to translate to the clinic as soon as we can. We feel that that's an easier, it's a goal that's more easily achieved, and it's also a goal that's more appealing to the FDA. So if you take somebody who's 40 who has hearing loss in both ears, and we know prognostically based on the type of hearing loss that when they're 60, they're going to have profound hearing loss unless something's done. And we have something that we can introduce into one ear, not both, just one, that we predict would prevent the hearing loss. We think that that's something that the FDA might find appealing.
So that's the route you're going now.
That's the route we're going
Okay.
right
But-
So are you looking for people with hearing loss in both ears to be a good?
now. Are you volunteering? Well, we should run you on those.
It's getting louder and louder, having the phone and the TV.
I would certainly be interested in volunteering.
Yeah, no, we can run you on OtoSCOPE if you'd like, not a problem.
So the history of him and I, especially as kids, were just a lot of tubes and he had ear infections. Does that have anything to do with this? Okay. Because Grandma and Grandpa were so unique.
So I'm gonna, I unfortunately have to go to give a talk at 1, but Kayla and Kevin are gonna stay here to answer all your questions, as well as other scientists from the lab. You can talk to anybody you want, they all know a lot of stuff, and we're all here to help you.
I just have, well, I'm not, Kayla can, I think there's a difference between me, and I think I'm 1, and my sister,
5,
and my 5, and then Helen, who is number
4.
4. Because Helen lost, it was a lot more dramatic when she got older.
Well,
And
I
mine-
always wonder about the external factors, because Donna and I, they found right away, they put these really powerful hearing aids on it, and we just got more deaf right away. And then with Helen had a really noisy animal clinic, and we just wonder if she lost hers faster because of the outside noise.
Yeah, but Jim's around
The
farmers.
But
tractor.
then you're around the tractors.
So I don't know.
That's true.
We actually are, so noise-induced hearing loss is very complicated, and it's likely complex genetic, and we're just starting a collaboration with a large group in Sweden, trying to use these technologies to solve part of the puzzle of noise-induced hearing loss. And some of the questions that you're asking, scientists couldn't have answered even if they wanted to 5 years ago, because we didn't have the technology to generate the sequence, and then we didn't, if we had generated it, we didn't have the bioinformatics to analyze it, and we actually do now.
You came out, made a comment that by 2020 there would be over 1 billion people with, and what makes you determine that?
So it wasn't me. It was a paper that was published, and that is actually based on as the life expectancy increase, which means we have more and more aging population, and a lot of proportion of this 1 billion is going to be due to age-related hearing loss. So, yes,
Well, the reason
yeah.
I brought that up because I was wondering if there might be some external factors there too, because I read
Yes.
an article about a lot of people wearing ear pods.
Yes.
And
Oh,
they
yeah,
said-
definitely,
And they
yes.
said that there's a big cause.
Yes, so that the billion, it's a hearing loss. It's not, we are not talking about genetic hearing loss. So it's genetic, but it's also, a lot of them is going to be environmental. It's noise-induced hearing loss. You see kids more and more wearing this huge thing, like you pass down the street and you can still hear them with their things. That's really bad. So they're not protecting their hearing. The other one is the aging population. With the advancement in medicine and all the treatments and therapies, people live like older and older, and that is just due to their age. It's just age-related hearing loss. But this is one, it's going to be a huge burden in healthcare. You can imagine 1 billion people having hearing loss. So that is why it is $1 billion. So the way I actually, yes. So I just want to finish to say that your family is actually, it was a complex study, as we showed you, but it was a challenging but in a nice way. Because we would not advance and we would not evolve if we are not challenged. Because we need to challenge our current knowledge, our old knowledge. If we don't, we still stagnant and there is no evolution. And your family is actually helped us moving with this whole thing. It was a very nice challenge. It is, when we look at your family and the hearing loss in your family, we look at it as a book. And we read so many chapters, but there are still many more to read and to understand. And that is what with the modifiers that Richard Kevin talked about, that is really, really new era in genetics. It's not just the genetics of hearing loss, that is any kind of genetic disease. It is, you look around you, in some families, some are more affected, any disease that don't have hearing loss. Some are more affected than others. Some, as you said yourself, start their symptoms earlier than others. Some go their whole life without developing any kind of phenotype or any kind of symptom. And that is such a great source for understanding more, discovering more. What does it make it better? What does it make it worse? And that's how we can actually intervene and correct or prevent. Mostly prevent, but we are moving forward to actually correct too. And it has been exciting. We don't have that many families that present that many variabilities, and we are really, really excited. And I want to say that it has been a journey working with you and in your family, and it was an exciting, and it's still an exciting one. So really, really, we are happy for you and to be so involved. Not that many families are so involved and so motivated and so passionate to know what is going on and to push, actually, which we really like too. We like being pushed. It's nice. It just makes us thrive more and more for like, yeah, that's a challenge. We can do it. And just seeing you and seeing like smiles, that for us, like, it means the world. It means it was worth it. So thank you.
So as more genes get, I guess, identified, is there plans to continue to compare our genealogy against those known?
Oh, definitely, yes. Because we choose what are the best candidates. These are the ones that are already involved in hearing. Because the probability that it is going to influence the degree of hearing is more, it's bigger than some other unknown gene that has never been seen with deafness. So that's what with your family, actually, as you saw, we screened with 4 different versions of OtoSCOPE. And we are going to screen it again and we screened with whole exome, like we went another level upper. Actually, the next step would be to do a whole genome. As I showed you, this is like the whole planet, so the solar system. That we are going to look at everything, not just the genes, everything in between, everything inside, everything out, trying to figure out what is going on. And again, it's a huge new concept in genetics and in human genetics, or even like all genetics, is this modifier. Sometimes it's not even in the sequence. So what we showed you here is kind of a lower complexity because we are looking for specific DNA, specific mutation. These are physical kind of. The other level is epigenetics. And what that means is the sequence of your DNA, we might sequence you. And the sequence is normal, it's completely normal. What is not normal on that sequence is what we call methylation. It's some kind of other chemicals that is attached to the DNA. And these are, you all heard about cancer. Yeah. And cancer sometimes it's not just, there are no mutation, it's just depending on these little chemicals attached to the DNA that will switch on or off a gene. Even a gene is completely normal. If it upstream of it there is a switch on, that should be off, or switch off, that should be on, that completely dysregulates the whole system. And this has been shown a lot for cancer, but it hasn't been really still very proven with genetic, what we call like genetic disease, like hearing loss, for example. So that is also another level. So what we are showing you is kind of different layers. So we kind of take one layer at a time and see if that makes sense or not. If it doesn't, we go to the next level. And that is like your family is one of the best to study.
Wow.
Yes?
I have a son, I'm not related to this family by blood, but I have a son with Meniere's disease. As you've done some of your studies, has anything come up in relation to genetic implications
Yes.
there?
So one of the projects we are working in the lab, another scientist is working in the lab, is actually Meniere's disease. And she has spent years trying to figure out what is the genetic cause of hearing disease, of Meniere's disease. We have several families from all over the world. Till now, no one really figured out what is the genetic cause of Meniere's disease, because there is a lot of variability, again, in the severity. There is also some genetic as well as non-genetic factors, but you are welcome. I mean, he is welcome to participate in the study if he would like to, and your whole family too.
What would he need to do? He lives in California.
So we can send him kits, saliva kit that he can send, or he can get his blood drawn somewhere, and they can send us any clinic, any hospital, and they can send us this. Or like we can, it's better to have a blood sample because the DNA quality when we extract from blood sample is better for the high throughput because we need a lot of it, yes. But yeah, both ways, either way is great. But we have a huge project in the lab with Meniere's disease, yeah.
Okay, thank you.
Yeah, my pleasure. So are you excited?
This is awesome. I'm so glad in my lifetime you came up with this discovery. I'm just in awe of you all.
It's a great, it is a great,
Yeah.
it is fantastic.
Okay, I have one more.
Yes,
The
please.
dominant, you're going to, and I understand this very well, that you're going to really put a lot of effort in trying to regenerate or gene therapy with the dominant hearing, yeah. But you still are going to, like with these mice
Yes, so,
experiments, you're gonna still work on the recessive and
yes,
the TCM?
yes, yep. So it is easier to work with the dominant.
Right.
Because with the dominant, you still have one copy that is functional, that is great.
Mhm.
So it is only about switching off the mutant one.
Right.
So it is easier to switch off one copy. The other one is still there, it is great. For recessive, you have two copies. You have to correct them both.
I see. Yeah.
That's why it is more difficult. It's not impossible, it's just a little more difficult and it will take a little longer time to get done.
Would any of the findings that you find with the dominant help with the recessive?
Yes. So anything, that's why it's like,
Yeah.
the family, your family and a few others give us the unique insights. They are really unique into the underlying genetic. It makes us understand even more molecular biology, the cause and the molecular level, biological level. It's not just about hearing loss anymore. It's way bigger than hearing loss. It's just about genetics of any kind of genetic disease. So that is why it is really, really exciting.
Ayla, could you speak a little bit, so you've talked a lot about prevention with the gene therapy. What about regeneration of folks that have, everything has died off and there's been a bunch of atrophy to the cells?
So a lot of people and a lot of labs, even here at the university, are working in hair cell regeneration. And there are some successes, but again, it's like about with science, we learn from our mistakes because we do make mistakes, but we learn from them. And there are a lot of unknown, but I think the road is going to be a little longer, but hopefully, I don't know how long, I cannot really, but we will get there. We will get there. So it is, I want you all to leave with like a note of a positive note. very optimistic, but yeah.
So going home today with kits for our family, is that a possibility?
Sorry?
Going home today with kits for our family to do saliva tests, is that a possibility?
Yes,
Okay.
yes, yep.
But
If
you
we
said-
can go to the lab,
Okay.
up to the lab, because we need consent forms.
Okay.
Any kind of research, we do have consent form. But other than that, yeah, Amy actually will help with that. Just on time.
Or did you say blood tests are better though?
So for the high throughput, like when you run whole exome sequencing and otoscope, it is better to have DNA from blood.
Blood.
But for genetic testing, it's targeted, we only, like if we get your DNA, we are not going to look at the whole exome or the whole genome. We are only going to look for TMCI, that specific exome where the mutation is. So for that, we don't need that much
So
DNA.
saliva is okay.
So, Amy actually brought some, Amy, you brought some saliva?
I have some saliva
Yep.
collection kits and sent for
So,
us.
yeah. So anytime, yes.
So if my husband and 2 kids are at home, just take them home and send
Yes.
them back? Okay.
Yes. Yep. Yeah. That easy, yes. It became that easy, yeah.
Yeah.
Is there any possibility we can get a copy of the slide presentation?
Yes, yes.
And we're also filming this too, so we'll make sure that you guys get copies of this. This has actually been, this is live, and this is on CNN right now.
That's a great.
Yeah, right. So Hale, is there anything else you'd recommend for the family to do other than if there hasn't been sequencing done to get the saliva testing done, but anything else that you'd recommend for them?
Actually, the usual recommendation is stay away from noise. Protect your ears, your kids' ears. That applies to anyone and everyone. Besides that, keep the motivation up. And please do not hesitate to contact us if you have any questions, any concerns, anything.
I didn't do an audiogram the last time. Would it be helpful if you had one? Because the rest of the family had their audiogram.
It would be great, yes, it would be great. We are planning on, as Kevin mentioned earlier with TMCI, with the splice site, that specific type of splice site, and one of the hypotheses is that there isn't the proportion between the mutant and the normal allele may be different, and that's what makes 2 of you have more severe hearing loss than others. So for those, we are kind of designing some experiments to see if that is true, and that we might contact you, or if you are interested, to give us some samples that we can kind of quantify the quantity of mRNAs and see if that actually holds, if this hypothesis holds.
When you had the audiograms up there, there was like a dark blue line that was below, at least on my curve, and I just wondered if that was a later, and it was a little bit below on Helen's. Yeah, you see on the left, I'm number 5. I don't see
Here,
it.
yes. So here, because I should have explained, so what is in here is every different color or line represents different age.
Right.
So here they may not be the same age. And it is an audiogram it's kind of more the average.
Okay.
Then so it is like a minor difference.
Okay, but the lower one there, is that the most recent?
This is the most recent we have.
We've lost.
So if you do have any more recent audiograms, we would love to have them.
How old are the other ones?
I think this one is in the 40 something. I'm sorry, it doesn't really show.
That was 2002.
Yeah, it doesn't show in there.
I think that was 2 years ago when we
I
were
was
here.
gonna say, didn't you
Last-
just have
last
them?
time.
Maybe that
Last
was
time we
2
got
years.
together?
Yeah,
2 years or 3
Maybe
years.
Yeah,
that
because
was-
you guys all went over to the clinic and had
That's
your
the one
audiograms done.
Helen-
Yeah, yeah.
Okay.
Jim, didn't you go over to the clinic?
Yeah, I did.
Yeah,
That's
you
right.
did.
the 4 of
Yeah.
us went over there.
Yeah.
Yes.
I have to say this. I'm a little bit disappointed. I mean, I learned today that the genes that the 4 kids have been identified. But it seemed to me, and Jack doesn't carry those genes, but it seems to me that that was kind of obvious from the start. I'm comforted by the fact that the grandchildren don't show carrying these genes, but concerned by the fact that if they marry someone who's a carrier, they have a 25% chance of their progeny carrying the genes.
Yes.
And I guess I was expecting more results from Dr. Gantz's prediction that eventually they will be able to regenerate the auditory nerve and recreate the cochlea. And I didn't hear much progress in that area today.
Yes, yeah.
I'm just wondering, what is the next step? And when, if ever, can our kids ever expect to have improvement on their hearing loss?
So for your kids, there isn't any right now. We cannot improve their hearing loss. Not anytime soon. The second about the regeneration, so it's not the auditory nerve in the case of TMCI. It's actually the hair cells that I showed. It's actually the problem is with the hair cells themselves. So the hair cells, that's what we need to hear. So the auditory nerve is a different thing. We cannot regenerate the auditory nerve. What we are trying to regenerate is the hair cells themselves. Because the auditory nerve could be intact. If the hair cells are not there, there is no regeneration, and there is nothing that it cannot be treated. As I said earlier, for hair cell regeneration, the road is still long. We are, like, it's not just us. We are working on it, and at the university, not especially our lab, because our lab is a genetic lab. So there are more biology labs that are working really hard on finding methodology to regenerate hair cells, but also all over the world. And till now, no one was able to do it except on chicken and some other lower animals, but not in humans. But doesn't mean they are stopped. They are still working on it, because that, again, regenerating, that's a huge step forward if we could do that. And everyone is really working hard. But as I said, it's about what we know right now. Everything is related to what we know right now. Our knowledge evolves, again, and we might change the way we look at stuff, we might improve it, and that's how it evolves and that's how we work. But right now, it was possible to regenerate hair cells in chicken and some birds, but not in a human. But we are getting there. That is nothing is, I mean, just to look as someone said earlier, just to look in our lifetime that we could sequence the whole genome. And it's not just my lifetime. So for example, when I started my grad school, I used to do it when I showed you that little thing that we screened exome by exome, like county by county in Iowa. I spent all of my PhD doing that. I was here every night, every weekend, because it took so long, and it was so laborious and so hard to do it one at a time. Now, and it's not, I can't even talk about my lifetime. In less than 5 years, I just submit something, and a month later, I have the whole genome in my fingertips. That is, for me, I'm still blown away by that. I still cannot believe in my lifetime that was possible. So it is advancing. Sometimes there is this leap forward. Sometimes it's just working, stumbling around, and then another leap. But that's science. That's knowledge, actually. It's not just science. That's knowledge. So.
Thank you.
Thank you.
I got a question. If you go down to the generation below, I guess that Tricia, that inherited my mom's mutation. That's a 1 in 9 million or something like that?
That's
9 in 1 million.
No, 1 million.
9 in 1 million.
Yes, getting there.
And so if they would match up with somebody that mutated like Dad's, which is 1 in 100, their chances are
Very slim.
higher than.
Oh, chances not to have, yes. So, this is for these two to meet. So it's all about probability. So 9 in a million would carry this mutation. One of you would be carrying this one. So you kind of multiply this by this. That is your chance
Okay,
to
but
meeting someone and then to having a kid that had both mutations.
so if you have the mutation that's 9 in a million, then you're just up against the 1 in 1,200 chance of matching up. So who is it in the family that has carrying my mom's?
I think Max and Ray did.
All of you. Oh, for the grandkids?
For the
Yeah, for the
grandkids.
Yes.
Max and Ray had moms, and then my kids had dads.
And we don't
Max
know.
and Ray is in more.
But I could
Yeah.
be either, is that right?
Yes.
I haven't been
Yes,
tested.
you will be either, yes.
Max and Ray
But I
would
will be
more
one
kids?
or the other.
No.
Yeah.
No, that's-
100 would be
But
more
just one,
kids.
the other one is completely normal.
Okay.
So you have nothing to worry about yourself.
Okay,
Yes.
Yeah.
John's,
who's carrying mom mutation? Max and Ray.
Max and Ray.
So they have a greater chance.
no,
No,
No, it'd be the other way around.
But
they-
Who has
they're
that
9 out of
mutation
a
has a
million.
bigger chance,
But
right?
they
Yes.
have the mutation.
Yes.
Yes.
If they meet that one person out of 1,200 that has dad's
So
mutation,
that's why we're concerned about Aaron.
No, we're
With
more,
dads.
but we'd be more concerned about them
Yeah.
finding the 1 in 1,200 to match the rare
Yeah,
one.
that's true,
Yeah.
yeah.
so Max and
Just
Ray
have
are.
them all get their
little more.
tests in and
We'll
get
get it
their own
tested.
genetic.
Yeah,
yeah.
On
We
the
were
sec-
concerned
So on
about
the,
the
I've got a
900.
solution. On the second date, they have to be genetic.
exactly.
Their second date will be, their second date will be in our lab.
Exactly. You know, it's like when I called up, what was I gonna do? Even if you had, I wasn't going to.
But
Yeah.
You know, I
you're
was
saying
gonna-
that both of these are recessive, correct?
Yes.
So, so in my case, I've got the one in blue, I would have to have somebody else in blue,
Which is
to-
bigger.
then that would cause that to proliferate?
No, you can have someone in blue or in red,
I see.
or some other mutations, but the same gene.
Yeah, it's fine.
It has to be the same gene, so the chances are really low, are very low. Yeah. So, it's kind of you got lucky in a certain way because it could have been dominant.
Yeah.
And then if it were dominant, it means if you have one or the other, you would have hearing loss. Yes.
So when I post this on Facebook, first, we didn't pay for this study, and so we want to thank you for all the.
Our pleasure.
So I posted on Facebook, and somebody says, I wonder about my hearing loss. Is that open to other people that ask us and say, is it possible to send in a genetic sample to the University of Iowa and have them look at it?
Yes. So we do offer OtoSCOPE as a diagnostic test, as a clinical diagnostic test. So you can go to the university, to the hospital, and they will order the test. If it is your family, you are welcome anytime. For other people outside of the family, it's also straightforward, and it takes a couple of months to get their results back.
And do they have to pay for it?
Yes.
Yes, probably.
Yeah.
Some insurance will pay for the test. Just depends.
Okay.
Is it important, even if we just have one of them and it's not really affecting us as grandkids? Do our ENTs need to know this for any reason?
It's one of those things that more information is always better.
Okay.
It's not, this information as it stands today is not going to change any of their treatment or decisions they make on you in the next 10, 12, 15 years in the clinic.
Okay.
So, but it's always better, more information is